Hi, my name is Melanie Philofsky, and I am hosting the second annual lightning talks of the fourth annual OHDSI Symposium. This year we have 8 speakers, and they're each going to give a 7-minute talk. We're not going to have any Q&A after the talks, but please meet up with everyone during the reception afterwards. How do I get it to move over? All right, there we go. Our first speaker is Iannis Drakos. He's the Chief Consultant of Research and Precision Medicine, Operations, Research and Innovation, Region Zealand in Denmark. Currently, he is leading the design and implementation of precision medicine in Region Zealand as part of the National Vision for Precision Medicine. He is also the project leader for integrating omics with clinical data for the European Society of Clinical Cell Analysis.
Thank you. Hello, everybody. I'm very excited to be here to describe a journey that began in Denmark 3 months ago. So we are relatively new to the OHDSI community. And the main reason that I'm here is to transmit the message to anyone else who is also new and having concerns if this is the right way to go, that yes, it is the right way to go. In Denmark, we are having multiple efforts targeting the utilization of the rates data scene of health data to promote precision medicine and in general tools for the bedside. And in this effort, during summer 2018, everybody in the country who was working in the field created an alliance so we can do the evaluation and tests together. I have to mention the Danish region of Copenhagen, the region of Zealand, Copenhagen University, the University Hospitals of Denmark, and a special mention to Odysseus Data Services, Inc. But without their help, we couldn't achieve the speed and smooth level of integration that we couldn't even imagine when we began this effort. So to you understand what we are doing, I have to spend the time describing the health data scene, which has a unique person identifier going back to 5 decades, late 60s. And in Denmark, by law, every bit of health information produced within any level of healthcare system, from primary care to hospital, is digitally stored and archived. And all this information have this unique identifier and can be cross-linked together. After being stored to the National Archive, they are distributed in 116 quality databases, and this process includes intensive quality control and governance. And anyone involved in clinical practice or research can request to have access to this data. It has to go through a strict application process. And there is also a nice data life cycle, and at any level, if there is an error identified in this data, it will be, the correction will be submitted back to the national registries and even to the electronic health record that holds the original information. And just to give you an understanding, I'm having a list of our registries. Some of them are starting from 1875, the Causes of Death Registry, or the Cancer Registry, which began during the Second World War. And the point is not to go through all the registries, but to identify or to describe the richness of the data scene in Denmark. So what we are trying to do now within the precision medicine is to convert this information and give something back to the people who produce it, and either help citizens not become patients or patients go back to being citizens as soon as possible. And for this, obviously, we need a strong research infrastructure that will accelerate the process of converting research results into clinical services. And we are focusing on improving the healthcare services and having on top data and security by the legislation being extremely strict in the country of Denmark. So within the National Vision for Personalized Medicine, that is an investment running and will continue running for the next 5 years, we did a lot of traveling, going around the planet to identify the best integration layer So out of these 116 national registries that we have, to be able to combine this information and to make it more easily accessible to researchers, but also clinicians. And during this evaluation process, traveling around the world, we identify many, not many, but some data models. And following what is happening in Germany, in the Netherlands, in Sweden, in Switzerland, France, and United Kingdom, we were convinced that the OMOP CDM was the way to go. And actually, we were surprised to see that the German Ministry of Health even referring to OMOP CDM by name in their guidelines for health data storage. So before making the final commitment, that became official yesterday, so now it's not an evaluation anymore. We will go with OMOP CDM as the foundation for precision medicine in Denmark. We choose 6 characteristic sources as colorectal cancer, biochemistry, pathology, drugs, anesthesia, and microbiology to build a proof of concept and convince everyone, including our politicians, that this is the right way to go. And these are the numbers. I don't have time to go into the details, but we are using around 20,000 different concepts to describe 75,000 patients. And this CDM and the tools became live, so in production a few weeks ago. That's why I'm saying I'm here to describe the journey. The results are not so concrete yet. Even though we have nice overlap between the different domains contributing information and the 75,000 subjects we are having. And the lead research in this effort is the Clinical Center for Surgical Science, led by Professor Ismail Goganur. And we are targeting to see how we can help patients with colorectal cancer reduce postoperative complications, the reappearance of the disease, and reduce mortality. And at this point, I would like to invite anyone who feels brave enough to collaborate, some people who are in the beginning of their journey, to collaborate with us and run studies that will help us better understand OHDSI OMOP CDM and the community tools, but also will help others interact with the Danish data scene. And this is some very preliminary results, but what happened around 10 days ago, and even though it's the first results, the quality indexes are very promising in regards predicting postoperative outcomes for colorectal cancer. But what I would like to focus is how this began. And it was during June 2018 when all the stakeholders of personalized medicine in Denmark gathered in a room. And the main reason that we were convinced to continue with OMOP CDM is the way these researchers are looking to Peter from Erasmus Medical Center. And we were very lucky and fortunate to have Peter and Patrick visiting us, explaining how we can do things and what exactly is the benefit by using OHDSI community tools. And they were discussing with Jesper Grarup, is the person that you see on the left, and Ismail Goganur is the person that you see on the right. This discussion was in June 2018. And less than 3 months afterwards, we have newspaper articles describing what Jesper is doing in personalized medicine. And this happened yesterday. It's one of the biggest events in Denmark about innovation, business, and health. It includes more than 5,500 participants, and as you can see, the Prime Minister gave the welcome speech. And we had 2 invited speaks from our administration describing exactly the benefits of OMOP CDM, because the preliminary results, at least at administration level, are quite impressive. And I can tell you about Professor Ismail Goganur that is in the process of writing, is being accepted, and in less than 2 months it will be an editorial in a surgical journal with a nice impact factor. It's between 6 and 7. Describing the benefits of using OMOP CDM to accelerate the results of research into benefits for patients. And this is a big thank you to everybody of you, because the biggest score in our evaluation wasn't the advantages of the CDM that was identified as the most advanced, wasn't the tools that offer the more integrated services, but it was the community. And I can say this with confidence because I'm part of similar communities, even with code contribution on GitHub, and OHDSI is by far the best community I've joined. Thank you.
Our next speaker is KP Kim. He's a medical oncologist and clinical pharmacologist by training and practice. His work includes precision medicine, PH1 clinics, and GI oncology. He is very involved with the OHDSI Oncology and Genomics Work Group and leads the effort to add genomic data to the CDM.
Thank you, Melanie, for the kind introduction, and really thank you for the committee for inviting me to give a talk and share my experience on how we wanted to translate really precision medicine into something that would care for the patients. So if you look at precision medicine, actually, we think it's quite similar to the OMOP CDM in that it contains various levels, population, patient level. And not only that, but as I will show, other specimen levels and those kinds of levels all together and how you're going to integrate all these data to really have an impact on the care of patients. Actually, as precision medicine comes into the era, populations are not the one size fits all or the blockbuster era, but subpopulation stratification, different treatment protocols according to the patient status, and more to the predictive analytics. In 2017, March, actually, the Korean government did a big step in that they decided to nationally reimburse next-gen sequencing for cancer patients. That did have a very big impact, and for us right now, too, we're sequencing around 50 patients a week. So the amount of data was just accumulating too fast. But we really wanted to have this table or box that we could really contain all this information and have a better impact on patients. But we were also understanding some other points. As next-gen sequencing was entering real world, we did see some differences from when we did it as a research-based study. So when it was researched, the sampling or the procedures were quite homogeneous. It was quite operationable, and you didn't have to think about other things. But with it in the real-world data, it was quite heterogeneous. It was you had to be really flexible on how you're going to obtain the data and really put it into, link it with clinical data, and how you're going to compare with prior Sanger sequencings or immunohistochemistry data. These were the various things we had to think about, what was the source, when was the next-gen sequencing performed, what was the consent status, and various other assays that were on board. How are you going to all integrate that? But at the end of the day, everything was centered around the specimen, and really, as they say, tissue is always the issue. So if you look at the tables, when we put in the database relationships, we would put the specimen, patients, and a procedure that was put in to obtain these specimens. Was it an operation? Was it a biopsy? And those kinds of things would mix together. And through the genomic meta table, we would put them into basically 4 tables. For single nucleotide variants or indels, we would put it in the mutational table. For copy number variations, we would put it in there. And for translocations or fusion genes, we would put in the structural variants. And for expression, for RNA or protein immunohistochemistry data, we would all put them into the expression. And with these 4 tables, we thought that we would be able to address all these various status of genomic data. As you can see, we built a DB, and I guess at the end of the day, the important thing was that specimen was in the center, and everything was centered around that. And I guess these tables, I won't go through every line of it, but you can just see that they were just basically on how we're going to get the data. And as you can see on the last column, we put in optional. So of course, in the really perfect world or in the research world, you could obtain all these data. However, in the clinical world, depending on vendor, depending on each the Department of Pathology, there was a variation on which information could be put into the table and which could not be obtained. And Expression was another very important part, we thought, because really how are you going to mix this up together with your prior molecular pathology data was very important to see that it was very consistent across the board. This is a diagram of the data flow of our institute, and as you can see, we are very fortunate that we had the OCS EMR imaging system and tissue bank, REDCap, and the registry data, and genomic lab information system all together, mixed up, and then we would send ETL them all to the data warehouse where they would go on to the CDM, and through the CDM we could be able to capture these clinical phenotypes and genomic data. Some extra issues we did have was, so for these data, we wanted to really go beyond just collecting data and really have an implication in everyday practice. So we wanted to link it with the tissue banks and build an API so that we could build further applications that could be used for precision medicine. This is just an internal validation data that shows that the genomic data that was captured by the CDM and our clinical database, that was the GLIS, there was showed identity. And for external validation, we chose two things. So for TCGA, we used it so that we can see that really is the genomic landscape similar. The diagram on the left shows colon cancer, and the right shows lung cancer, and easily colon cancer, as expected, was quite similar, while lung cancer had an East Asian flavor to it that differed from TCGA with higher incidence of EGFR. Another thing we did was to see how we were doing operationally, and an institute, Bundang Seoul National, really helped us out on that part, and to see how the data was captured within their sequence and really how it could be, that the tables were quite nicely functional. We went on to build applications to really see how this would get, how we could easily identify it at the population level. At the patient level, you might not be able to see, but really how treatment was put in, which was first-line regimens, what was second-line regimens, and what other immunohistochemistry data were all on board. And then for next-gen sequencing, sometimes you would do a re-biopsy or another next-gen sequencing again, so how that would compare what was concordant, what was discordant, and at the specimen level too. For the tissue bank, for this tissue, is there plasma, or is there buffy coat, or is there normal tissue, or tumor tissue, and what was the quantity of it? You could all just easily confirm. These were some of the points we had to think, consents and other cancer CDM specific issues that we think should be more evaluated by the community. And these are my acknowledgements. Thank you very much.
Next, we have Dr. Mary Regina Boland, an assistant professor of informatics at the University of Pennsylvania. She is a leading informatics researcher in the field of prenatal and perinatal exposures and has studied the effects of birth seasonality on later risk of disease in several populations, including the Americas and Asia, where she presented as a lightning talk at last year's symposium. Dr. Boland has also confirmed some of her findings in humans in a canine population. She is currently expanding her work into European populations and is looking for more OHDSI collaborators interested in her work.
Thank you, Melanie, and thank you all for being here to listen to my work. So the first, I wanted to start with a question, and this question, which I think many of you may know the answer to here as clinicians and terminology experts. You may know this answer to this question. So what do these two things have in common? This would be a motorcycle accident and a pregnancy. Does anybody know? Possibly? Perhaps? Okay, so these are actually both V23 codes in ICD-9 terminologies, and the only difference is the version. And you can detect this actually by, if you don't know this, you can detect this by seeing a dramatic shift in your gender population over years, as women are commonly diagnosed with pregnancy and males are commonly diagnosed with motorcycle accidents, which I found very interesting. So this always made me laugh, actually. So this actually highlights the importance of linking terminologies to standard structures that can then be mapped across multiple different terminologies, not just different terminologies, but different versions of terminologies. So as a member of OHDSI, they have used the SNOMED CT, which is the systemized nomenclature of medicine, and also expanded upon that to map all of the different codes in different terminologies to this standard architecture. So this is just a schematic of how different codes in different terminologies can be linked to different SNOMED concepts. And SNOMED concepts are actually organized into hierarchies, so then you can leverage those hierarchies, which I'm going to talk about in a few minutes. But this became important as part of my birth season project, because initially we started out, and this is the map here, the purple diamonds showing the different sites that we had originally. And when we tried to add data from the Netherlands, which is a collaboration with Peter Rijnbeck and those at Erasmus Medical Center, this kind of highlighted some of these interesting issues with the terminology mapping. So the first part is that when you have, and this, I'm sorry, this got garbled, so this must be some issue with the Mac to PC thing. So it's worked on Melanie's laptop, and we checked, but unfortunately, it's a PC here. But this issue is the first column with there's about 354 condition concepts that were common in the US sites that were part of this study. So this included Vanderbilt University, University of Washington, New York City. We had Columbia University as well as Mount Sinai data. And there were 354 condition concept IDs that were common across these sites. And these are, we restricted it to have at least 1,000 patients per condition concept. So there are some prevalence constraints as well here. But when we started to add the 2 Asian sites as well as the site from Netherlands, if we add all 6, or 7 actually I think sites, we end up with only 701 condition concept IDs that are in exact complete agreement, whereas the exact same concept ID in all sites. So what we did was we then started to look at, so we have a total of 4,340 condition concept IDs that are unique if you were to pool all of this data together. And then we were able to, using the condition concept maps, we're going to focus a little bit on the Netherlands data set. So there were 249 unique Netherlands condition concept IDs, I should say, that were unique in their set, which means that they were not found in the other sites. So the first thing we did was we used this concept hierarchy to map them to other concepts that were found at other sites that were close within the hierarchy. So this is the concept map kind of from SNOMED. The big numbers on the far left-hand side, so 0, 1, 2, 3, 4, 5, are the different levels in the concept. So 0 being kind of the top level, and then 5 being a lower level, so a more granular concept. So what ended up happening was Rotterdam, which is in the Netherlands, mapped things to the congenital anomaly of the cardiovascular system, whereas Nashville, New York City, Seattle, and as well as the Taipei group, mapped to congenital heart disease. And these are actually 2 levels apart in the hierarchy, which is very interesting. And just without knowing that this is the hierarchy, I would have thought congenital heart disease is more of a vague term than, say, congenital anomaly of cardiovascular system, so I probably would have organized it the other way. But you have to remember that this isn't necessarily semantics or other types of things. This is kind of just the hierarchy, how someone designed it initially when they designed SNOMED. They put these different levels here. So that's another important thing. And what we can do is we can actually roll up or move up the hierarchy and kind of map things to kind of more general concepts if we need to. And another interesting example is this genuine stress incontinence. In the US, there seemed to be a lot of this kind of gender focus where they had female stress incontinence, whereas at Rotterdam they used a mixture of genuine stress incontinence as well as mixed urinary incontinence, and there was no I guess if you wanted female stress incontinence, you'd probably combine a stress incontinence code plus the gender code, but they didn't actually explicitly say this is female stress incontinence, which happened in some of these other sites. So I show this graph because most of the concepts could actually be mapped. If you mapped 2 levels of separation away, you could then actually map quite a lot of concepts. So what does that mean? So coming back to the congenital anomaly example, that would mean mapping everything that's at the 2 levels lower, so which would be like congenital heart disease up to congenital anomaly of cardiovascular system. And you can do this, and this is kind of the approach that we're sort of looking at taking. However, I do kind of want to caution people, and I think this is also important, that a lot of these inconsistencies still kind of do have to be manually reviewed, and this is a little bit of an unfortunate part of this process, but because some of the semantics within the hierarchy are a little bit vague, and you do have to kind of dig into why was this originally mapped to this concept at a particular source terminology, this is an important caveat of these data. I do want to thank the data sets originally, as well as my collaborators on this work, Heinrich and Peter, and thank you very much.
Next is Fabrício Kury, a medical doctor from the Federal University of Rio de Janeiro, Brazil. As a postdoc at the U.S. National Library of Medicine, he performed clinical studies using EHR data and Medicare claims data, as well as analysis of medical terminologies. Currently, he is at Columbia University, where he focuses on bridging observational research to clinical trials.
Thank you, Melanie, and thank you, the Symposium, for the opportunity to talk. This study, I hope you accept as a kind of invitation. It is one of those things that needs to be done, will be done, so shall we get started? We know that we can do studies that produce medical evidence analogous to clinical trials. In fact, 3 years ago, I did one of those studies, and personally, I was impressed by how similar the results from my study were to the results of the paper from the journal Critical Care that I was replicating. So what I want to call attention to here is, what about we take this paradigm of study replication and we apply to clinicaltrials.gov? In other words, what if we took studies that are currently recruiting patients and tried to predict the results before the study gets completed? To that end, I tried to contribute a bit of, let's say, legwork. I tried to better understand how feasible would it be to satisfy the data requirements of the clinical trial by means of an electronic phenotype that would be executed against an imaginary stereotypical repository of patient records. So I took from where, I'm sorry, there's something with the slides. I took from where Amy Wong and colleagues left off at the AMIA Symposium last year. In her model, she classifies the data requirements, how feasible it is to query for something. The easy things are the structured data. The hard things are those that you need to manually review the chart of the patient to know. And I added a bit of nuance on top of that. I'm sorry it was supposed to show here, but I added the notion of the presence. A parallel notion to how easy it is to query is how likely, how compatible to the standard of care is what you're trying to find. An available thing is something that is commonly done in clinical practice, and an unavailable thing is, say, an exam that has no justification as per clinical care, or a drug that is not yet in the market. So using this, let me try to briefly show how this works. These are two studies from clinicaltrials.gov. In the bottom one, quickly, the intervention is the invitation for screening for hypertension. Now, even if that happens, in regular clinical practice, it is unlikely that you're going to record in the patient's chart that the patient was invited to screen for hypertension. So that's considered something that is not present, regardless of how hard would it be to query for that. So with that in mind, I appraised myself 648 individual data elements from the 25 largest phase 4, currently recruiting, interventional studies in clinicaltrials.gov that do not involve placebo and have at least 2 study arms. A universe of 1,600 studies. And this is how the results look like. 1 in 7 data elements, not trials, was hard. And 1 in 5 was not available. The studies had on average 25 data elements each. And most of them had at least 1 data element of each type, but not all of them. And here, I am not considering the possibility that you can use a surrogate for a piece of data that you don't have, or that you can decide to ignore a piece of the study because you deem that it is not critical for answering the core research question of the study. Out of the 25 studies that I appraised, I found 5 that I consider to be readily reproducible using observational data. Let me quickly show you 3 of those 5. In the top one, the question is, what is the best duration for antiplatelet therapy in acute coronary syndrome as measured by all-cause mortality? In the middle one, the question is, which is the best anticoagulant for patients with atrial fibrillation? And in the bottom one, the question is whether the choice of drug for general anesthesia influences the cognitive ability of the patient 10 years after a surgery. Now, just to comment about this very quickly, one can argue that, for example, all-cause mortality might be hard to follow up on, but perhaps not in all countries. And the cognitive ability can be hard to query for, but the mini-mental state examination is performed, and it is a keyword next to a number. Perhaps you can try to make an electronic phenotype for that. So I'm not trying to claim that the appraisals of one person, me, is a gold standard, or 25 trials are representative of 1,600. I'm just trying to claim that the outlook is the opposite of bleak. So in order to propose a conversation about this and help organize it just a bit, and taking from where Amy Wang left off in her paper last year, I think it's useful to draw a distinction between the presence and what I call the queryability of a data requirement, even if just because those 2 problems have different solutions. The presence you can try to address by reaching for privileged data sets, say a hospital specialized in a type of condition. And the queryability you can try to address by developing electronic phenotypes. So if you want to see examples of things that are hard to find or hard to query for, please visit the GitHub repository with all the results, line by line, the data elements. And I just want to finish with one last fact, which is 619 clinical trials recruiting patients right now at an average cost of $20 million per trial. How nice, to say the minimum, would it be to try to predict the results of at least some of those studies? We are talking maybe tens, maybe hundreds of them. And even if the results don't match, if the predictions don't match the actual results, what lessons are there for us to learn by giving an earnest try? Thank you.
James Weaver has a background in epidemiology, statistics, and data science with experience in academia and the pharmaceutical industry. Currently, he is the manager of epidemiology analytics at Janssen Research and Development, where he designs and executes population-level effect estimation studies for effectiveness, safety, and methodological development. Jamie has contributed statistical code to the OHDSI codebase, helped develop an algorithm to link mothers, babies for perinatal research, and has collaborated in network studies. And this year, he is faculty in the population level effect estimation advanced tutorial.
Thank you. Hi, thanks everyone for having me. This lightning talk is on the value of negative controls for the self-controlled case series design. One of the many purposes, or a main purpose of OHDSI rather, is to generate reliable evidence for the benefit of patients, providers, researchers, healthcare systems, industry, and government agencies. One method for ensuring reliability of evidence generated using observational data is to execute one study against negative controls. The purpose of this talk is to support the use of negative controls in observational research and show an example of how negative controls can characterize error and uncertainty in a study. And to that end, we replicated this study published earlier this year in the New England Journal of Medicine that found a 6.05-fold increased risk of primary inpatient myocardial infarction in the 7 days following laboratory-confirmed influenza diagnosis using the self-controlled case series design. We suspected a health services utilization bias might be responsible for at least some of this effect, namely that being sick enough with the flu to seek medical attention is temporally associated with all manner of short-term follow-up care, and not necessarily just MI. This is a population-level effect estimation study, which is used for identifying causal relationships. It attempts to estimate the unbiased average effect of an exposure on an outcome. It does so by comparing outcome occurrence between comparable populations, and causal effect identification can be done provided it meets consistency, positivity, and exchangeability criteria. The self-controlled case series design is a method for doing this. It compares outcomes within persons during time periods of different exposure status. It meets the consistency criteria by using precise time-stamped risk intervals and outcome events. Positivity by requiring that patients in the models include both exposed and unexposed time. And the exchangeability criteria is met by considering the unexposed time as the counterfactual approximation to the exposed time within the same person. Because you're balancing within people, this ensures confounding control for time invariant or mostly time invariant factors like sex, genetics, and lifestyle, but it does not ensure balance for time varying confounders like age or seasonalities or factors that are correlated with this. For our replication, we specified it in various ways. Our exposure was influenza, and one approach was to use a diagnostic code for the flu. Another approach was to use a positive lab result for the flu. We ascertained the flu in the outpatient visit setting or in the any visit setting. The risk interval was in the 7 days following the influenza diagnosis, and our outcomes were primary position, first occurrence of myocardial infarction, and 31 negative controls. We executed a simple self-control case series analytic variant, and then we included 3 others that are meant to account for violations in the assumptions on which the self-control case series design relies. Here are the 31 negative control outcomes that we used in this study. They include conditions such as lung cancer and HIV and schizophrenia and, of course, ingrowing nail, none of which are caused by the flu. So here are the results from the New England Journal paper put onto a calibration plot, where the x-axis shows the magnitude of the effect, the y-axis shows the magnitude of the standard error, and the dashed diagonal line shows a effect to standard error threshold where the nominal p-value equals 0.05. Items below this line are statistically significant at the less than 0.05 level, and those above are not statistically significant. The paper reported the 6.05 increased risk of acute MI following the flu, and they used one negative control outcome and reported a 1.35 effect that was not statistically significant. Our results, these are the results from the simple self-control case series analysis variant, where we found a 4.85 increased risk of MI following the flu, and all of the vast majority of our negative control outcomes were showed similar positive effects, with extreme variability of effects from 1 to 12. The suspected health services utilization bias is supported by these results. This variant was when influenza was captured as a diagnostic code in any visit, and the outcome was in the inpatient setting. If we vary the outcome definition to include any visit, many of the negative control effects are still positive, although not all of them are positive, but the extreme variability in the effects is maintained, where they're between 0.5 and 7. The suspected health services utilization bias may be less subject to outpatient visits, which make up the majority of these outcome events. If we vary the influenza definition to require lab confirmation, we see that the effects are in the same direction for when the outcomes are defined in the inpatient setting and in the any visit setting. We just have less statistical power to, as you can see in this plot. So really, negative controls are an important diagnostic to understand the validity of study findings, and this applies to self-controlled designs as well. This shows that negative controls can be used to identify problems and to quantify error, test hypotheses, and compare alternative methods. But what they cannot do is solve the problem that they've helped identify and generate new hypotheses. Using this method, the self-controlled case series method, under various different specifications, we've been unable to find a specification that puts all the negative controls at a null value. So we're going to try different study designs moving forward. Thank you. I'd like to thank all the people on this list, especially Jeff Kwong and company at the University of Toronto, who have been nice collaborators in trying to get to the true answer of this question.
All right, next up is Clair Blacketer. She's a member of the Epi Analytics team at Janssen Research and Development. Her research interests include prediction modeling and data integrity standards. As a member of OHDSI, she's the project manager for the Common Data Model Working Group.
All right, good afternoon. As Melanie mentioned, my name is Clair Blacketer, and I'm going to be taking you through our study looking at bleeding risk prediction models. So when we think of a serious condition like bleeding, it's really important that a patient know their risk of developing a major bleed, particularly when considering beginning a new drug regimen. So luckily for us, there actually have been models created to calculate this risk. And what we wanted to look at during the course of this study were how good are these models that are currently out there, and can we do any better? So as Jenna showed us this morning, when we approach a prediction problem, we want to really frame it up in the OHDSI way. And how that looked for us was, amongst new users of warfarin or direct oral anticoagulants with a prior history of non-valvular atrial fibrillation, bit of a mouthful, but that's our T, who among those will develop the outcome of a major bleeding event, our O, within a year of the start of treatment? And that is our time at risk. So given that this is the problem we were interested in, we decided to choose some existing risk models that also looked at similar questions. So these were Atria, Hasbled, and Orbit. And we also decided we wanted to look at CHADS2 and CHADS2 VASC. And now you're probably telling me in your head, Claire, you're wrong. CHADS was developed for ischemic stroke. You are right, but there actually has been some research out there that shows that it's also predictive of major bleeding events as well. So in order to answer the question, how well did these risk scores actually work, we applied the 5 scores to the target populations across 4 large US claims databases. And then to answer the question, how can we do any better, we then learned a regularized lasso logistic regression model on those same target populations across the same 4 databases. And what that allowed us to do was then compare the performance of the existing models and our learned models. So if we focus specifically on the model we learned on the Optum DoD database, predicting major bleeds in new users of warfarin, we had a target population of about 132,000 patients, of whom 3,700 went on to develop the outcome. Within that target population, we fed LASSO about 109,000 different covariates that we wanted it to consider in our final model. So if you look at this graph here, the x-axis shows the proportion of those covariates in patients without the outcome, and the y-axis shows the proportion of the baseline covariates in patients with the outcome. So any of these dots, each one representing a covariate that falls above this 45-degree line here, would be considered a positive predictor, and any dot falling below that 45-degree line is considered a negative predictor. So if we think back to our risk scores that we're interested in, for example, HAS-BLED, they started out with about 20 to 30 covariates, and they ended up choosing 9 that you can see here, many of which that fall along that 45-degree line, so indicating to us that maybe they're not the most predictive, and clearly there's some room for improvement here. If we look at the additional 4 models we were interested in, they had similar performance in terms of their covariates and where they fell. So the question then becomes, what covariates did the LASSO algorithm choose? So of our 109,000 covariates, the LASSO algorithm chose 455. And yes, that is a little bit more complicated than those models that have between 5 and 9 covariates. But what comes with that complexity is the ability for us to use so much more of the data that we have at hand. So now the burning question is, how did this actually work? So if we take a look at the ROC curves, oh, it didn't really show up right. If we take a look, there we go, at the ROC curves for our existing risk scores here, you can see that they achieve about an average AUC of 0.66, with HAS-BLED performing the best at a 0.68. Our PLP model achieved an AUC of 0.75. Looking at some specific points along these ROC curves, for example, if we wanted to look at a sensitivity of 50%, meaning that we are identifying 50% of the outcomes, we still are achieving a nice specificity of 83%, whereas HAS-BLED is achieving a specificity of 65%. More typically, we want to look at how our model performs at the high specificity level, where we only have a 1% false positive rate. And we can achieve that and still find about 8% of our outcomes, whereas HAS-BLED is only finding less than 1% of our outcomes. Digging into our model a little bit more, you can see our ROC curve again here on the left and our nice calibration plot here on the right. And one of the major benefits to using the PLP framework is that we were able to take that Optum warfarin model and apply it to the additional 3 databases we were interested in, giving us a nice external validation. And you can see the model still holds up, even in these disparate populations. And so it really gives us confidence that our model is robust. So in the end, what we found is that our PLP models that we developed using the patient-level prediction package consistently outperformed the risk scores that are used in clinical practice. And this really lends further credence to the argument that the PLP framework is really how we should be approaching all prediction problems in the future. Thank you.
Next is Ross Williams. He is a PhD student in the Health Data Science group working at Erasmus in the Netherlands. His focus is on prediction modeling, specifically method development for model transportability assessment. He contributes to the OHDSI patient-level prediction workgroup and software package.
Great. So I'd like to start off by saying, firstly, thank you for having me along to talk today. I'm talking about predicting 1-year risk of heart failure for patients with type 2 diabetes. So if you were listening to Jenna Reps this morning, you'll recognize the time at risk, the outcome, and the target that we're looking for. So why did we choose this problem? So firstly, it's a very clinically significant problem, because if you have type 2 diabetes and you get diagnosed with heart failure, you've got a 50% 5-year mortality rate. One of the other things that we also noticed is that in the guidelines, there are already recommendations. So for instance, with I'm not even going to try and pronounce that, with TZDs, their use of caution is cautioned in high-risk patients. However, when we were doing a literature search, we were unable to find any viable prediction models for this problem. So if I say you caution when you're at high risk, but you have no way of saying that a patient is at high risk or not, there's not really an effective way to use this guideline. So clearly we have a need for a well-calibrated, externally validated prediction model. Now, the problem definition. So if you've seen any of the PLP work, you'll have seen this diagram. So for us, our target population is what we call pharmaceutically treated diabetes. Our outcome is the heart failure, and our time at risk is for 1 year. So to dive a little bit more deeply into these cohorts, so we say pharmaceutically treated because we want our index date to be where we envision this model being used. And that's when, as a clinician, you're sat with a patient and you say that their diabetes has progressed to such a level that you need to make a pharmaceutical intervention. We're no longer going to try and do it with sort of lifestyle cures or lifestyle amendations. We're going to use a drug. And on the day that you make that drug, we want to say, what is the risk of you developing heart failure within 1 year? These cohort definitions are all available online at the link at the bottom. One thing to note is that we allowed people to have a diagnosis code up to 365 days after the event. This is just an artifact of how some of the data goes through the systems. For heart failure, so we just use a condition occurrence and an inpatient visit. However, in some of the databases we looked at, due to their nature, so for instance, the IPSI database in the Netherlands doesn't contain information on hospitalization. So we have 2 definitions of heart failure, one with the hospitalization and one without the hospitalization. Both of these are available at the links below if you want to find out more. Now, how do you do a patient-level prediction problem? If you want to do a patient-level prediction problem, your first step is look at this paper that's been written. This tells you everything that you need to do. It's the best practice that we have available. So how did we move from our clinical problem that we've set up into making this into a prediction exercise? So we developed multiple algorithms, including GBMs, lasso, and random forest. I'm only going to talk about the gradient boosting machine results here because they were the best that we achieved. And then we used the following covariates for our model development. Now, we had 11 databases that were available for this. These span, I think, 6 countries and 4 continents. And as you can see, we've got like quite a large number of patients, so 1.7 million, 2 million, and lots of outcomes. What we were really concerned with here is external validation. So our method of doing this was, in each of the databases, we'd develop a method, and then we would validate it in the other 10. So that's what you can see from the diagram at the top. And then what we did is we rotated the development databases. So you start in database 1, and then you validate in the other 10, and then you start in database 2, and you validate in the other 10, until eventually what you get is this diagram. And this is all the results. So to notice here, so we have the validation database along the x-axis, and the development database is along the y-axis. So if we move to look at the lead diagonal, what you'll see here is the internal validation scores. So if I select one of these squares, so this is Panther, this is the model developed in Optum Panther that we have then externally validated in the data that we have available in CCAE. And so the performance is great. And so any of these squares, you look to the left, you'll see the development database, and then you look down and you'll see what database it was validated in. The scale on this, so the red is 0.5 in area under receiver operating curve, which is basically a coin toss, and then 1, which is the bright green, is your best performing. So if we select the model that we think is the best performing, which is the Optum Panther one, you can see that we've got good to excellent performance. And not only that, but it's maintained across these data sources. And one thing to say is that not only are these from different countries and different populations, but they're also from different database types. So we have some electronic health records in there, we have some GP databases in there, and we also have claims databases in there. So what we think is this model is very robust, very transportable, can have a direct impact on clinical practice. One other key thing to note is that we know that calibration is important. So if you have a well-calibrated model, it will have a better clinical impact than a model with similar discriminatory performance, but a worse calibration. Actually, this is one of the new flexible calibration curves that we've just added into the package. If you're interested in this, you can come and see my poster, which is poster 27 in the next section. But to conclude, what I would say is that we have very good model performance here. So model performance between sort of high 70s to 90. This is preserved across multiple data sources, and we've also got excellent calibration on here. And so I feel like this study really helps demonstrate the power that the OHDSI community can have in producing these prediction models and giving the confidence to clinicians to actually use these prediction models. And we can do this at a global scale. And I would say that to do sort of 7 of these database validations, it took me 1 day. So that's something to remember when you are running these studies. And the other thing that's nice is that if you are a data stakeholder and you have your data in the OMOP CDM and you want to add a line to the diagram that I have, you can go to this GitHub link, you can download it, and you'll run the best performing model that I created against your own database. So thank you very much for listening to my talk.
Next, we have Kristin Feeney Kostka. She's a data science lead for Converge Health, which is a division of Deloitte Consulting. She specializes in clinical data science for real-world evidence generation and translational research. Kristin's most recent work focuses exclusively on using artificial intelligence to better clarify patterns of disease. Since 2015, Kristin is the self-appointed and still uncontested Global OHDSI lead for Deloitte involvement. She is also a perennial member of the Symposium Planning Committee.
Thank you, Melanie. So, I have a confession to make. I actually plagiarized this presentation from all of you here in this room. We are part of a village, and by sitting in this room, you are now a member. I'm going to tell you a bit about our tribal history and a little bit about the process that we have that you may not even realize how innovative it is. So I want to ask you a question. How long is too long? How short is too short to do research? Don't go somewhere else with this question. I have to disclaim, I'm here wearing my OHDSI hat, and I am a human, so therefore I am totally likely to be biased in what I'm about to present to you. And I've already drank the Kool-Aid, so if you're not already part of this community, well, join me in this journey. We have some benchmarks on what normal looks like. And it's a Sisyphean task, our favorite, right? It's like rolling a rock uphill. Our friends at the Sentinel Initiative have done a great job showing us what we can do when we move to a simple descriptive analysis or a complex descriptive analysis all the way through to an L2 inferential analysis. And the time on average that they reported in some of their benchmarks was 50 days, with the majority of that being spent on study planning. Now, to move L1 and L2 in total, the range gets even longer. And these are for somewhat comparable tasks, somewhat comparable drugs. It just shows you that we spend a lot of time thinking. But there really is a point of diminishing return. Why do we do this to ourselves? In economics, this is a popular thing to show up, but for some reason, we forget this in clinical research. I want to challenge us to think about breaking our problems into smaller problems. Because when we think of things in software development, like the waterfall to agile, we can break these down and we can accomplish things in shorter amounts of time. We did this year, some of you know. We hosted the face-to-face study-a-thon. We came around this idea of generating reliable evidence for one clinical problem. Our colleague Bridget Wang presented this on a community call. There's a wonderful amount of information about it. I'm not going to talk about that science. I'm going to talk about all of the pieces that came together and the process that we adhered to. Because a study on studies is just as important as the study itself. I know that sounds a little bit like I said the word study 300 times, but wait for it. It gets better. We take for granted that there is quality, accuracy, and reproducibility in this process. And we need a better job of documenting the baseline perception of what normal is today as we continue to evolve this community. So I did just that. I turned my lab of people at Columbia into a cohort that I studied. And I took some baseline perceptions. Some of you answered this survey. It was a pre-test. As they started the study-a-thon exercise, as they went off and marched into their groups, whether they were T, C, O, negative controls, we wanted to know the average time to complete an observational study that they had done previously, huge range of time. And we see that on average, at least half of that is spent on planning. Is that too much time? Is that enough time? I don't have a great answer, but I challenge you to think about it as we go through this. There was some confidence in the quality of the study, and a little more in the expertise of their colleagues. And it was shared across the board. The representativeness, the robustness, the reliability, somewhat confident in something that might have taken 1,200 days of their life. Seems a little crazy. After the 2-day exercise where we went heads down and answered that question, I surveyed these people again to see if maybe they felt a little differently. And I actually did get people to consent to this so I could report on their information. We had 8 people who reported this was the first time they ever did this. Maybe I shouldn't have asked them their opinion in the first place, but it's good to know that we had newcomers. We see even after completing an exercise like this, 75% of the people in that cohort still could barely agree on what a reasonable amount of time was, even though they had been part of an exercise to turn it on its head. We see that 12 researchers attempted to execute the study package, and attempt is a pretty important word here. And I will come back to this in a moment, but it's important we acknowledge it's an attempt. And as you might expect, people actually felt pretty confident about robustness, repeatability, having the expertise on hand. And why is that? Probably because they knew there was more data available to them than they might have in their own house. We can see that in general, people were very likely to want to recommend this to other people to do. But they also acknowledged they might not be able to do it where they are today. They might need to work on some change management to influence others who aren't as new to this process. What are the key takeaways? Well, I'm just going to be totally honest. We didn't actually finish. I finished my study on studies, but some of the artifacts of the other study to be completed. And maybe that's okay. Maybe 48 hours is a little too aggressive to go from study conceptualization to publishing in New England Journal of Medicine. But Patrick, I still think you're right. I still think it's doable. I mean, look at those negative controls, man. We could do this. But we need to do more characterization of the contributing data sources. Andrew is hosting a great data quality workshop. Maybe you're signed up. Maybe you're going to sneak in. But people are challenging the data that contributes to these studies. But we know that there are axes here that we're measuring on, and we can see comparable evidence. My colleagues before me presented wonderful examples of larger populations consuming less resources. I believe Ross just said it took them a day. A day! How incredible is that? I remember watching code run in grad school and being happy with 30 minutes of like one line of code running. So I give you credit, Ross. I'm positive you had more than one line. And I will totally admit that we are already in a place of becoming where we believe in these ideas. And so I might just be surveying people who already bought into the change. But great minds tend to think alike. If you haven't seen this, you should. Our friends at Flatiron did something very similar and came to similar conclusions. 48 hours was too short a time for them. They did a couple of months, but they came to a similar point. Our villages are starting to redefine the paradigm, and it's because of villages like ours that we are moving this forward. So thank you to everyone who is doing this, because the process is just as important as the research.
All right, that concludes our lightning talks for this afternoon. Another round of applause for everybody that came up and spoke. I encourage everyone to stay for the networking reception and meet up with these speakers to collaborate and further the work of OHDSI. Thank you.